Good afternoon, everyone. I am Tanveer Abbas, student of BS Poultry Science at PMS Arid Agriculture University, Rawalpindi. I would like to thank the organizer and for the opportunity and all of you for joining me today. Uh, as this is a time of lunch, so I will try to make it short and digestible, just like snacks. The topic of my presentation is bibliometric analysis of the fowl adenoviruses from 1976 to 2023. My contents of discussions, introductions, objectives, introduction, objectives, methodology, resultant discussion, strength and limitations, and then conclusion. Fowl adenoviruses are the DNA viruses. They are causes heavy economic losses to the poultry industry. The major structural proteins of the fowl adenoviruses are uh, fiber 1, fiber 2, penton, and hexon. Penton and fiber 1, fiber 2 are related to the entry or the attachment of virus to the host cell, and hexon is related to the specificity of the serotypes. It is classified into 5 species: A1 to AE, FADV A1 to FADV A, and 12 different serotypes that are FADV 1 to FADV 8A and FADV 8B to FADV 11. They are causes diseases like inclusion body hepatitis, hepatitis hydropericardium syndrome, and adenoviral gizzard erosion. What is the bibliometric analysis? Is a systematic study of a scientific literature. It uses quantitative methods to know the contribution of the influential authors, institutes, journals. As much of the studies has been conducted on their structure, on their pathogenicity, epidemiology, characterization. So our objective was to conduct a bibliometric analysis to evaluate the scientific contribution by leading author, journal, countries, and institutes, as well as publication trends, explore collaboration, and learn about the research trends and hotspots. Methodology, we have collected the data from a Scopus database, and our search string look like this. We have included 628 articles and 24 reviews. Data analysis, several key metrics were used to assess the performance, including the total publications, citation per publications, co-authorship, bibliographic coupling, occurrence and co-occurrence of keywords, H, G, and M indexes. H index measures the productivity and citations. G index gives weight to highly cited articles, and M index is the modification of the H index. It gives the academic length. The researcher remains active divided by the H index. Different tools were used. Wasviewer, Microsoft Excel, Biblioshiny. Now the results and the discussions, publication and citation trends as is depicted in the picture. It rises, the total publications rises and reached their peaks at 2022 with 65 in numbers. Total citations and H index reaches their peak in 2018. Now the most cited articles, they are the most cited articles, and in the top 2, 3 or 4 reviews are mentioned. It suggests that reviews gather more citations than the articles, and Michael Hess appears more in the most cited articles. Michael Hess from the University of Veterinary Medicine, Vienna. Analysis of the sources. Avian diseases ranked first in terms of total publications, while in terms of total citation per publication, H index and D index, avian pathology ranked first. Analysis of the authors. Michael Hiss from the University of Veterinary Medicine, Vienna, contributed a lot by publishing 53 articles, either a sole author or in collaboration. He ranked first in total publication, total citation, citation per publication, H index, and G index. Eva Nege from the University of Gulf, uh, next to the Michael Hiss with 37 publications, and 1.83 M index for some author from the Yangzhou University, Chinese Academy of Agriculture Sciences, shows that they are the emerging authors. Now this graph shows a comparison of the publications, total publications of the authors and the publications of authors in the recent era from 2019 to 2023. Some of the authors, Yi Jiangqiang, Shao Hangzia, Qin Eijan, Viking Wang, they have their total publication in the recent era, suggests that they are the emerging authors. And co-authorship analysis basically examine the collaborations of the authors. So this is conducted on the VAS viewer, and it gives clusters. Each cluster is look like a team that is based at any institute. There is no minimal or no collaboration between these groups. Results and discussions, analysis of the countries. China has produced more publications than any other related to the FADBs. Co-authorship analysis of the countries. Maximum collaboration was found between the USA and Australia, Malaysia and Nigeria, Austria and Hungary. Overlay visualization describes the earlier work came from countries, and the recent work came from Iran, Egypt, Malaysia, Nigeria, and China. Analysis of the institutes. University of Gulf produces more publication than Shandong Agricultural University, University of Veterinary Medicine, Vienna, Yangzhou University, Chinese Academy of Agriculture Sciences, Free University of Berlin, Henan Agriculture University, National Veterinary Research Institute, Pulau, China Agriculture University. They are the top contributing institutes. So this is a temporal breakdown which shows three eras: 1976 to 2000, 2001 to 2014, and 2015 to 2023. Only University of Gulf is present in all these three eras. In first eras, Free University of Berlin and Commonwealth Scientific Industrial Research Organization remain dominant. In second era, University of Gulf and University of Veterinary Medicine, Vienna remain dominant. And in the last or the recent era, Chinese Academy remain dominant. Bibliographic coupling is a science mapping technique based on the assumption that two publications sharing common references are likely to exhibit similar content. So we extract the titles of each cluster, and we know that most of the work has been done on FTD4 or hepatitis hydropericardin syndrome, and adenoviral gizzard erosion is less focused. Author keyword occurrence and co-occurrence analysis shows the most occurred word that shows the current state of the research or established research. Fowl adenoviruses, hepatitis hydropericardin syndrome, inclusion body hepatitis, pathology, hexon protein, vaccine, phylogenetic analysis, PCR, fiber protein, adenoviral gizzard erosion, and epidemiology are the current state of research. The less connected word or least occurred word that are the co-infection, RT-PCR, CRISPR-Cas9, isolation, Leghorn, male hepatocyte cells, RNA sequence, apoptosis detection, indirect viruses, monoclonal antibodies. They are the less connected word and shows that much of the work should be, they have to be explored. Strength and the limitations. This is the first bibliometric study on the fowl adenoviruses, and it highlights the collaborative network, research hotspots, and key contributors for guiding new researchers and policymakers. limitations, data from Scopus database only, citation biases that are self-citation, review papers, novelty bias, keyword limitation, 139 paper lacked relevant keyword, no gray literature, only English publications included. Future studies should integrate multiple database, improve keyword inclusion, and address citation biases. Finally, conclusion, a bibliometric analysis shows 10.4% annual growth of the documents related to the FADV's research peaking in 2022. China is the most productive and influential country, while the USA leads in collaboration. Key contributors, Michael Hess from the University of Veterinary Medicine, Vienna, Ivan Nage from the University of Gulf, and emerging scientists from the Chinese University, Jian Qiangyi and Tofan Li. Top Institute, Zhengzhou University, Chinese Academy of Agriculture Sciences, and Shandong Agriculture University are driving recent advancement. Top journal, Avian Pathology. There is limited collaboration between the authors based at different countries. So limited authorship, research teams work independently with big global collaboration. Emerging countries, China, Iran, Egypt, Malaysia, and Nigeria are increasing their research contribution. Research teams, bibliographic coupling highlights established research, while less connected and less accurate keyword reveal research gaps and hotspots. Encourage international collaboration to strengthen research impact. Expand multidisciplinary approaches by integrating virology, immunology, and vaccine development. Explore under-researched areas to address emerging challenges in FADVs. Improve keyword usage in publication to enhance discoverability in bibliometric studies. Thank you.